Thanks for having me here in such short notice. So really, yesterday I heard I had to come here. So apologies if I'm not well prepared, but I'll do my best. So we just thought it was very important to show the benefits of working with the IDDN cluster and other academics in Ireland to Sigmoid and to our pipeline. So let me see if I get this right. No? Yes. So I will just briefly, most of you know Sigmoid, but I will just briefly introduce us. Um, uh, David mentioned about one of our technologies, LEDs. I'm going to focus more in our latest development, that is the single multiple pill and all the benefits that we have from it. And we also have used the IDDN cluster to characterize and understand how this technology works. I will give three examples, one in poorly soluble drugs, one in delivery or peptides, and also in our oral vaccine program. And then I will conclude in all of this under 10 minutes, so bear with me. So we are based in DCU, in the event center. We are privately owned, and it was founded in 2003 by Ivan. We are around 20 employees, and Ivan has raised up to 13 million so far. Our core expertise is drug delivery technology development, and with the LEDs and the Simpil. And our lead product is the Cycle. That is a product that we are hoping to bring to the market in the next 4 years for the treatment of ulcerative colitis. And some of the preclinical work package that we use, for instance, to present this product to the FDA and to other regulatory agencies, was done through Cormac's group and other groups in Ireland that help us get that basic knowledge of the mechanistics of how this product works. And obviously, we have an extensive network of clinical academic industry, and we have a quite strong patent portfolio. So this really summarizes what we do at Sigmoil. So I want this. So in the upper area, we have kind of the cyclosporin-based indicate products that we are trying to develop. Our lead product is, as I mentioned earlier, the ulcerative colitis one. We have successfully completed a phase 2A, and we are now getting ready to a phase 2B. We plan to be in the market in 2017 with this, and we have 2 pivotal studies to do. So it's heavy times ahead of us, but we are quite motivated. Then we have the cyclosporin opportunity. Also, we believe we can play a role in the treatment of Crohn's and graft versus host disease and immune suppression in general. Cyclosporin is generally used for transplant patients so that they don't reject the organ, but it has never, there's no product on the market for the treatment of IBD. So it's really bringing a known drug with known toxicology to a new indication. So it's really a product that we developed in-house from concept, and we hope to continue to market. Then we have other in the pipeline. So with the Simpil, I will try to prove to you that it's broadly applicable across all classes of drugs. And in a few slides, I hope you'll understand why we say this. And then we have oral vaccine program and an oral peptide program, among others. But these are the ones that we hope to bring some we brought to the clinical, and these ones we hope to bring to the clinical in the next 2 years. So that's why they are here. So when we speak about drug delivery, if you think about the drugs that are coming, the new chemical entities, they generally are, everybody talks about these poorly permeable, poorly soluble. They come with a lot of issues, unstable. So really you need a drug delivery technology, or a number of them, to meet the different needs of these new chemical entities. And then, of course, you also have to target to the site of action, and you need to understand where there is. So one of the approaches that generally the companies in drug delivery take is they either do a top-down or a bottom-up. So the top-down, they basically, this is an example, is the nanocrystal technology at Elan. So basically you start with a bulk, like a crystal, and you just mill it down so that you have really fine particles. And then the other approach, normally people use solvents to solubilize the drug, and then through spray drying or those type of techniques, you just form very fine particles. But all of this, what you are getting is increased dissolution. So you're just getting a benefit from that is limited always by the solubility. So what we try to do with Simpil is we try to solubilize the drug and deliver it in the solubilized form. So it doesn't need to dissolve. It's already in the ready state to be taken. So that's really where we think we have an advantage to what is out there. We believe we address all these issues, solubility, permeability, stability, and release, collectively. And I will just explain a bit more in the next slide. So how do we address this collectively? Basically what we do, we use, of course, GRAS-listed a solution of API. So basically solubilize the API with our in-house know-how. Then we have this solution into a polymer matrix, and we make this liquid that is then brought through is pumped through a heated tubing, and when it's cooled down, you just form these nice spherical mini beads. But the beauty of this is through combining an hydrophobic API solution with a hydrophilic polymer, you basically create microenvironments within a 1.5 millimeter bead. And these microenvironments allow you to actually tailor where, like select where you want your active to be, where you want your functional excipient to be placed. So you can really play with your formulation from all angles. Then, of course, so this here is an example of how the Simpil looks like. And you can then, of course, coat them. You can even have different populations coated, some coated to one area of the GI tract, others coated to another. So really so that you tailor the treatment to what is needed, and you target the site of action. And as mentioned earlier, we do deliver the drug in a liquid form. So you don't need to be controlled by dissolution or anything. So now just a quick example. So the cyclosporin example here, I'm not going to talk about the lead product. It's just an example of what we can do with the technology. So what we are trying to show here, like cyclosporin is a very toxic drug. So you want to avoid to deliver more than you need, because that will just destroy your organs. So what we did here was, this is compared with Neoral. So this is Neoral, and this is the bioavailability of our Simpil cyclosporin. And what we did, if you look at the pharmacokinetics, the area under curve is equivalent, but you lose that toxic effect because all of what we deliver is within the therapeutic window. So you basically kind of have fit for purpose bioavailability. So this is really important, and the message here is we can tailor what we want to get from the formulation. In the peptides scenario, and this, as I mentioned earlier, we have done all of this in collaboration with David's group. We chose in the IDDN calcitonin because it's really it's a very difficult peptide to deliver. So it's very low permeability, and it's sensitive to heat. So there are a number of issues that you need, hurdles that you need to overcome to deliver it successfully. So here, after we used all the tools that David mentioned, so basically that allow us to select, let's say, permeation enhancers that would be successful with this peptide, allow us to select between formulations that would work and wouldn't work in vitro, and then bring that to oral scenario. This data is in this data we target the junon in rats. In rat model, and we did oral gavage of a number. We have here 3 formulations. And basically, we were very encouraged by the results, because if you look of the benchmarks, so in clinical studies in humans, you have less than 2.5%. This 1.5 is in preclinical in the same type of rat model, and we are getting 10% bioavailability. And even this formulation here, without any permeation enhancer, is showing to be better than the benchmark. So this is really promising, and this is opening doors to us to engage with other companies that have peptides that are difficult to formulate. So this is very useful for us. Our other program is in oral vaccines, and we have done this work with the group of Ed Lavelle in Trinity. So basically what we do, we formulate the drug, and then this is kind of what we do, we dose, this is all through oral dosing. So we dose the animal in days 0, then we do a boost on the day 13, and we take blood samples, and then we do another boost and we take blood samples, and then in the last day, on day 35, we just sacrifice the animal and we check, you know, all the levels in the blood, in the mucosa. And basically, this is just one example. We have loads of this. So this is just to show that after we immunize, here you have three formulations, 1, 2, and 3, with Simpil, and then compare with the solutions of the antigen and the antigen with a second antigen, just have a higher effect. And basically what we have here is that with using Simpil and then challenging these mice, we had 100% protection for one formulation. And basically we have tried this with a number, also bacteria subunits, but also whole kill cells and a range of others, and the data is pretty consistent. So really, in conclusion, and I won't go through this detail, is really just to say that we wouldn't have done any of this if we didn't have the network behind us to really prove the concepts and the mechanisms. And it is really very valuable to us. So thanks for listening.